Thanks very much, Stefan, and thanks to the organizers for letting me talk today. I'm going to talk to you about a very specific story here, and this is a discovery of a new gene with a striking role in neurodevelopmental disorders. And this is going a step back away from the therapeutics here and talking about a story of diagnostics. So one of the important things to note, if we're thinking about severe neurodevelopmental disorders, for example, we currently only find a diagnosis for around 40% of all patients. That limits our therapeutic approaches. We can only design genome-targeted therapies for those 40%. So there are 60% that we still don't find a diagnosis for. And these are figures from the Deciphering Developmental Disorders cohort from their sum up of 10 years of working on the project. But our approach to finding these diagnoses in patients is very heavily focused on the regions of the genome that directly encode proteins. Now, I will not ever pretend this is not a strategy that makes perfect sense. We can look at a very small amount of the genome, around 1.5% of it, and variants in these regions are likely to have a large effect. And crucially, that is an effect that we can predict. We have this absolutely beautiful amino acid code that allows us to say with quite specific certainty what the effect of any individual variant will be on the protein sequence. And this is really transformative for our ability to predict variant effect. But what about the variants outside of these protein coding regions? We kind of neglect them a lot in the diagnostic setting. And now, when we think about the genes across our genome, now we have about kind of 18 or 19,000 genes that are on the left-hand side of this figure that are transcribed into RNA and then translated into protein. These are our protein coding genes. But there are tens of thousands more genes that are actually transcribed into RNA and function specifically as RNA molecules. They're not translated into protein. And our approach to diagnostics, and when we do exome sequencing, we're very focused on the yellow side, the left-hand side of this figure, and we completely ignore everything on the right. Now, in the UK, we are incredibly fortunate that many years ago, our government invested in genome sequencing. And we've heard a lot already about the Genomics England project, and specifically the 100,000 Genomes project. And these data are transformative in our ability to look at variants in non-coding regions. So rather than just looking at those protein coding sequence, we now have data in the entire genome. Now, I'm going to tell you a specific story about what happens when we look outside of this. And this is a discovery made by a wonderful DPhil student in my team, Young Chen. He sat here at the front, so please accost him with questions afterwards. But he came across what looked initially as a very strange variant in the middle of one of these non-coding RNA genes called RNU4-2. Now, we found this, well, Yang found this variant in 46 individuals within the 100,000 Genomes project. And in 40 of these, we knew this variant existed only in a child and wasn't found in either of their unaffected parents. So they'd arisen de novo. Now, we normally expect the de novo variants are random occurrences. We expect them to be pretty scattered across the genome. So to find 46 individuals with the exact same variant, I will admit that I said to Yang, this is rubbish. Carry on. Let's move on. But he was somewhat persistent and said, well, hang on a minute. All of these individuals have undiagnosed neurodevelopmental disorders. And we don't find this in the kind of 50,000 odd individuals in Genomics England that have other phenotypes or have an existing diagnosis. Okay, fine. We should probably look into this in a bit more detail. So this actually transpired to be about 0.5% of all of the unrelated individuals with neurodevelopmental disorders in Genomics England 100,000 Genomes project. So here what I'm showing you is a plot of this gene. It's only 141 base pairs long. And this tower of triangles in the middle of the plot here is this variant, with each triangle being a different individual with a de novo occurrence of this specific variant. And what we actually noticed, the bottom half of this plot here is showing you variants found in the general population. So here it's the UK Biobank, 500,000 individuals in the UK Biobank cohort, where each dot here is an individual variant, and the length of the lollipop, so the stick on that dot, is the number of individuals that have that variant. But I want to point your attention to this red bar in the middle, where we see a depletion of variants in the general population. So there are fewer of these dots, and they have very tiny lollipop strings. And this is a position of where this variant exists. And if we look in this wider region in Genomics England, we find a few more variants. And when we collaborate with wonderful colleagues such as Anne O'Donnell-Laurier at the Broad Institute to identify additional individuals, we found very quickly, actually, 115 different individuals, all with neurodevelopmental disorders, with variants in this very small 18 base pair region of this non-coding RNA. And actually, quite strikingly, and it's interesting when we start to think about therapeutics here, actually about 3/4 of these individuals all have that exact same variant, which was the first variant that Yu Yang identified. So we collaborated with Anne and her team at the Broad, particularly Gabrielle and Sarah, clinicians in her team, to look at the phenotype associated with these variants. And we had 49 individuals in which we had very comprehensive phenotype information. So all of these individuals have global developmental delay, with this ranging from moderate to severe, but most of them being on the severe end of the spectrum. They all have intellectual disability. A large proportion of them have microcephaly, are short in stature. Most of them are nonverbal. They have hypertonia, issues with vision and feeding, and a large proportion of them also have seizures. And most of them have the involvement of multiple organ systems, so it's not just a restricted brain phenotype. And actually, through this story, I became very aware of the complexities of syndrome naming. But actually, in discussions with the families, they decided to name this disorder ReNU syndrome, which is somewhat of a positive name, which I will come back to the reasonings for this later on. So what I've told you is this is this non-coding RNA. It's 141 base pairs long, and it has an incredible role in neurodevelopmental disorders. Why? What does this thing do? Well, it's a key component in the major spliceosome. So if I take you back to your high school biology, DNA makes RNA makes protein, but after that first RNA is made, it comprises of both regions that go into the final transcript, whether that's the protein coding sequence or the final RNA, called exons, and regions that intersperse those exons, called introns, that need to be removed through this process called splicing. And RNU4-2 encodes the U4 small nuclear RNA that is part of the massive spliceosome complex of proteins and RNAs that are involved in mediating this process. And particularly, U4 helps to recognize the left-hand side, or the 5' end of the intron, and tells the spliceosome to splice in the correct place, so to remove the correct amount of intron. So this is just a cryo-EM structure of a part of the spliceosome, the so-called U4, U6, U5 tri-SNRP complex. And on the right-hand side here, I'm just showing you the U4 snRNA in red. And this is tightly wound to another small nuclear RNA, U6, shown here in blue. And these two things are complex together, and they need to be unwound in order for U6 to receive or directly bind to the 5' splice site for splicing to be activated and these introns to be removed. So if we zoom in one step further, so in both of these figures, I'm showing you the U4 snRNA in teal and the U6 in grey. And the position of the variants are shown by either arrows, where they represent single base insertion variants, or in orange if they represent small or single base changes. And these variants cluster to either side of this 18 base pair region, which is underlined on the left, into two structures. One is this stem 3 structure of base pairing between U4 and U6, and the other is this kind of weirdly named quasi-pseudoknot or T-loop structure. And what these two structures are thought to do is they accurately position on the left here this labeled U6 ACA-GAGA sequence, literally just sound out this DNA sequence. And that is the sequence that directly binds with the 5' splice site. So these structures hold that in the correct position for splicing to accurately happen. So in order to check that these variants are actually impacting this splicing process in this way, and particularly this 5' splice site selection, we looked into RNA sequencing data from 5 patients with variants in RNU4-2. And this was analysis done by Ruby Dawes, who's a wonderful postdoc in my team, and also Jenny Lord, who's now a lecturer in Sheffield. And we could show on the left here that in these cases with the RNU4-2 variants, they have an increased number of changes in 5' splice site usage. So instead of using the normal end of the intron, it shifts slightly. But the thing with what we're representing on the right here is that this is not random. So they don't just randomly use a different site. Actually, the same different sites are used in these individuals with the variants in this gene. So it's a systematic shift in 5' splice site usage. So just to join the circle a little bit here, so on the right here, what I'm showing you is this U6 ACA-GAGA sequence, which I mentioned earlier, and how it directly binds to the 5' splice site. And particularly, it binds to the so-called +3, +4, +5 positions of the 5' splice site. And on the left here, I'm showing you this shift in consensus sequence in the sites on the top of this diagram that are increased in usage in these individuals with RNU4-2 variants compared to the ones that are decreased on the bottom. And the difference in these two sequences are at those exact same 3 positions, the +3, the +4, and the +5. Now, that was quite a lot of data, but what I'm trying to show you here is that these variants impact 5' splice site selection in a way that is systematic. So they changed the usage away from the consensus normally used sites and used somewhere different. And this is a process that happens on a low level. It's quite a subtle signature, but a low level across different genes across the genome. Now, one of the slight complexities when we're thinking about non-coding RNAs is that the U4 snRNA is actually encoded by a whole bunch of different genes. There are actually 90 different genes across the genome that can encode U4. A bunch of these are pseudogenes, but actually RNU4-2 is the only one in which we find variants that cause these severe neurodevelopmental disorders. Why is this? Well, with Stefan and the wonderful Alicia and her team, we looked at data from the BrainBar database, and we show that RNU4-2 is actually the major brain isoform. It's expressed at a far higher level than the other U4 genes in the brain, and that's across development. So one of the things that was most striking about this discovery was the frequency. So most of the genes that are very common causes of neurodevelopment were discovered kind of a decade or so ago when we first had large-scale sequencing cohorts, or at least smaller family studies. But when we look across Genomics England, we find variants in the small 18 base pair region in 60 individuals. And this corresponds to about 0.7% of all of the undiagnosed NDD probands. This doesn't sound like a large number. But how does this transfer? It's quite difficult for us to put a number on how this corresponds to frequency in all-cause NDD. We don't have a whole genome sequenced cohort of individuals that are everybody presenting to clinic. But if we take the DDD cohort that I had that figure from my very first slide, and their figure of a 40% diagnostic rate, and assume a 40% diagnostic rate upstream of defining a gel undiagnosed NDD cohort, then this would estimate a frequency of 0.4% of all NDD caused by variants in this gene. Again, it doesn't sound like very many, but let's compare that to the frequency of the most high-hitting protein coding genes in this paper from the DDD cohort. Now, these numbers of 0.7% and lower are not much higher. However, the key difference here is that all three of these genes are protein coding genes in excess of 2,300 amino acids long. They have 7,000 base pairs of hit space for non-coding variants. And that compares to an 18 base pair region of 141 base pair non-coding RNA. I think that's very cool. What's also very cool is the difference that this makes to the families. So many of the families that we have now diagnosed with variants with ReNU syndrome have been on a very long diagnostic odyssey. So this is sometimes 8, 10 or more years of clinical appointments and different genetic tests. And many of them have communicated that they never thought they were going to get a diagnosis. On the left here, I'm showing you the ever-growing Facebook group. Every time I do this talk, I update my screenshot. The last time I checked, it had 175 different members in it. And my little thought experiment here is imagine the difference if you have a 3-year-old that's just been diagnosed, that it takes to meet somebody with an 18-year-old or a 20-year-old. You know what puberty looks like. You know your child's still going to be alive in that many years. We've also been working with Sarah Wynn and the Unique charity to try and join up and make a community out of these patients. I also just wanted to let one of the mums, one of the families speak for themselves of the impact that having a diagnosis has on their family. And I especially liked this quote because it hit me very hard as a computational person. So it's one thing to write papers and crunch all that data, then another is to see a family with a precious, unique child who is living it day to day. This is where data meets real life. And I told you I'd tell you a little bit about where that name ReNU syndrome came from towards the end of the talk. And this is where, in this quote, they said that they like to refer to RNU4-2 as ReNU because their family has been renewed by this information and hope for the future. So just to segue on that hope and actually join this up in the whole kind of therapeutic genomics space. So how do we take what is a striking diagnostic story and take that into a story of therapies? And I think we have a bit of an opportunity here to discover something that is so frequent at a time when we have all of these tools to make therapies is actually pretty exciting. But there's various different things that we don't understand and we need to understand much better before we can take this forward to therapies. So one of these is a question about mechanism. So I showed you the RNA-seq data for a reason, because we think the mechanism here is one of altered function. This RNA gets into the spliceosome and just makes it function differently. But we still don't understand what the effect of either increasing or decreasing expression of this RNA would be. We still need to understand more about the disease itself. When is its onset? How does it progress? So we're working with the families to look at natural history studies of this disease. And then our RNA-seq data at the moment is limited to blood, so we're working on making new cellular and mouse models to understand more about the biomarkers of this disease in a more appropriate cell type. And we've had some really exciting initial discussions, especially with Carlo, Stefan, and Matthew, as to how we might translate this finding into therapies in the future. I've skipped out a lot of the data that's in the paper, so if anybody is interested, then here is the reference. And it remains just to thank a cast of over 100 different people that were involved in this story. Particular thanks to Yang for being persistent and saying that we should carry on looking at this thing because it was super interesting. Anne and her team for help with the clinical side of the story, and Stefan and his team for help with the kind of the brain, the RNA-seq and the phenotyping side of the story. Susie and the team from GEL, who are complete superstars. And especially the patients and the clinicians and all of the recruiting centres for actually sharing these data and allowing us to make these kind of discoveries. And thank you very much for listening.